Of course, you don't now, hybridization-based or non-PCR based techniques of molecular marker, that is RFLP. RFLP restriction, RFLP stands for restriction fragment length polymorphism. That was first identified by the Botstein in a virus electron pneumonia. So if we talk about the RFLP is nothing but these are the genetic markers resulting from the variations of changes in the length of defined DNA fragment that is produced by the digestion of DNA sample. So we suppose we have a DNA strand here, A, B, C, D. There are different genes. In red side, you can see the restriction sites. If we are using particular restriction enzyme, if it produce the cut, it will produce different length fragments. Okay. So the polymorphism generated by the DNA fragment length is represented by the RFLP. So genetic marker, RFLP is a genetic marker that resulting from the variations or changes in the length of defined DNA fragments, which is produced by digestion of the DNA sample with restriction of DNA. The electrophoretic comparison of the size of defined restriction fragments that are from the genomic DNA. So how we can identify these restriction fragment, electrophoretic fragments? First, we have to isolate high quality of the DNA from a source organism, source plant cell. To digest with a combination of restriction enzymes. We can use individual combination of restriction enzymes. Okay. Then fractionate digestion sample by the electrophoresis. We have to run all these restriction fragments on the electrophoresis of the Jaegler cells. Then transfer the fragments to the membrane. We can transfer these fragments to the membrane. Hybridize with radioactively labeled DNA probe. Hybridize with radioactively labeled DNA probe and detect through photoradiography. Can also use non-radioactive labeling. So this is the representation of the RFLP. Here you can see agrogels, which is transferred to the nylon member through this Shutterbox Susan blotting methods. So later on, there photoradiography is only film can generate you can see clearly Susan blotting method. Detection of a specific DNA fragments from genomic DNA of an organism. So if we are studying DNA on the gel, that is known as Southern blot, because based on the name given by the E.D. Southern, that is the scientist E.D. Southern. If we are studying RNA on the gel, that is northern blotting, that is reverse to the DNA. So if we are transferring protein from the gel, okay, that is western blotting using antibody instead of different antibody. So this is RFLP analysis polymorphism revealed by different probe enzyme combination of 30 different axes. This is the RFLP bending pattern look like. If we are considering the use of RFLP for the relatively slow process, takes time, slow process. Use of radioactive isotopes has limited RFLP that is used to certify the laboratories, but non-radioactive labeling systems are allowed by the UG. It's a codominant marker of one species specific that need high quality of DNA, need to develop polymorphic probe also. It is somewhat expensive. If we talk about the application of this RFLP, it is at a specific level or among closely related taxa. Presence and absence of fragment that is resulting from change in the recognition site that are used for identifying the population. Estimating the genetic distances and fingerprinting. Estimation of genetic distances and fingerprinting. Forensic biological percentage paternity cases. We are using this RFLP. Disease status genetic mapping. PCR based technique. RPD, ISR, SR, FLP, ST, Scott models. So basically, PCR, as I told you, PCR polymerase chain reactions that was developed by the Kary Mullis in 1983 that bring molecular marker analysis to a new level. So the basic principle of molecular marker is nothing but it's a thermocycler. Okay, thermocycler. So the main objective of PCR is to amplify a particular DNA sequence into a multiple copy, number of copies. Basically is amplify the DNA sequences to the power n. N is the number of cycle, how much cycle we are. So it will produce a particular from one cycle it will produce two to the power n. One cycle two power means one to two, two to four, four to eight, eight to 16 like that, to the power n cycle. Okay. Now RAPD, randomly amplified polymorphic DNA. RAPD. RAPD are the genetic markers which resulting from the PCR amplification of genomic DNA sequences that is recognized by the 10 nucleotide number, 10 MERS, 10 primers. That is important. So in case of RAPD, RAPD stands for random amplified polymorphic DNA. Randomly we are amplifying. We do not have any fixed sequence. So randomly we have to design the primer. That primer act as a forward reverse primer, same primer, arbitrary nucleotide sequence. Okay. So these primer will act, same primer will act as a forward or reverse. It will randomly amplify the single effect. So RAPD are the dominant markers that require no prior knowledge of the any DNA sequences, which make them very suitable for the investigation of the species that are not well known. So in this case, we are designing arbitrary nucleotide primer. Arbitrary means without knowing the sequences, just randomly we have designed ATGC, GCTC for the 10 nucleotides. Okay. So luck by chance, these nucleotides will get bind, or get complementary binding, having complementary binding with that. So you can see here in the picture, A, B, here it is binding, it will get amplified. Here if you similarly here. So randomly it will bind to different locations in the genome and it will give you a different fragments. That is the basic the principle behind this RAPD, randomly amplification. Then we look for the polymorphism. So this is the RAPD profile it looks like. You can see. If random amplification is there, it will give you the random fragments. Now enter simple sequence repeat ISR. The generation of ISR markers involve PCR amplification of DNA PCR amplification, using a single primer that composed of microsatellite repeat sequence and in some case primer also contains 1 to 4 base anchor at either 3' or 5' or 4', which target a subset of the simple sequence repeats and amplify the region between two closely spaced or closely oriented SSR. So here the PCR amplification of DNA using a primer which have microsatellite repeated. So in this case, primer having the repeated unit. So at both sides, if we have repeated a unit, it will amplify the the region that is present between the repeated unit. Okay. Inter simple sequence repeat means both side simple sequence repeat sequence are there between inter. ISSR technique permits the detection of polymorphism in microsatellite and intermicrosatellite loci without previous knowledge of the DNA sequence. This is the diagrammatic representation. Now simple sequence repeat. Our DNA sequence will repeat length of few base pairs. Variation in the number of repeats can be detected with the PCR by developing primer for the conserved DNA sequences, flanking the SSR. This flank the SSR. As molecular markers, SR combine many desirable markers properties, which include high level of polymorphism and information content, which unambiguously designation of alleles, even dispersal, selective neutrality, high reproducibility, codominance, and rapid and simple genotypification. Microsatellite have become the molecular marker of choice for application. Application in genetic mapping and genome, genotype identification, varietal protections, varietal identification, seed purity evaluations, and germplasm conservation, diversity studies, paternity determinations, pedigree analysis, gene and quantitative trait locus analysis, marker study. All these are the application of microsatellite simple sequence repeats. So advantages: they require very little or not necessarily high quality data. highly polymorphic, evenly distributed throughout the genome, simple interpretation of the result, easily automated, allowing multiplexing, good analytical resolution, high reproducibility, codominant marker for new halogens. Codominant marker. These are the codominant markers. Then you can say it's new halogens. These are the advantages. Because they require very little or not necessarily high quality DNA. Because we are using molecular technology like PCR. PCR does not require high quality of the DNA. It can amplify if primary complementarity binding is there. It can amplify. So quality doesn't matter in case of PCR. Highly polymorphic, they are polymorphic, highly polymorphic you can say because they are repeat unit. Variation is because of repeating unit. If variation is repeat 6 times, 7 times, 8 times, so repeat unit determine the polymorphic of this marker. Evenly distributed throughout the genome, as you know, because the repeat unit of a particular are present throughout the genome. Simple interpretation, easily automated, allowing multiplexing. So this is the representation of figure representation of the SR molecular markers. So applications of the individual genotyping. Genotyping can be done by using this application. Parentage identification, parental identification, genetic diversity explorations, population genetic diet studies, population genetic studies, genome mapping, evolutionary studies, or hybridization. So can be identified by using these SR markers. Genome mapping can be done. Genome mapping can be done by genetic diversity, population genetic study also. Parentage identification, cumulative identification can be done. Next is AFLP, amplified fragment length polymorphism. So amplified fragment length polymorphism, here it's a combination of two techniques, that's PCR and RFLP. PCR polymerase chain reaction and RFLP restriction fragment length polymorphism. Here we have to amplify the restriction fragment of the DNA. Then we can able to develop AFLP, amplified fragment length polymorphism. Then we study the polymorphism. The informative fingerprints of amplified fragments are very important. So this leads to development of AFLP. So the AFLP technology is a DNA fingerprinting technique that combines RFLP and PCR. It combines these two technologies. It is based on the selective amplification of subset of the genome with restriction fragment using PCR. AFLP process protocol of AFLP, we have to digest the genomic DNA with restriction and the nucleogenesis. Then ligate the commercial adapters, different sequence at the both end. Here is the figure you can see clearly. I think it's clearly visible. This is the genetic material, DNA which we can digest by restriction enzyme. Here we can ligate the adapters. Carry out the PCR on the adapter ligated mixture using primer that targets the adapters, but they have very basic primer. So in this case, they carry out the PCR by using the primers that is complementary to the ligate sequence or adapter sequences. This is the AFLP profiling. So this is very sensitive. AFL very sensitive. Good reproducibility, but technically demanding. Relatively expensive technology because as PCR involved, only the technology involved. Discriminating homozygotes from the heterozygotes. You can easily discriminate the help of this AFLP. Require band quantitations, comparison of pixel density, and all. Because it produces large number of the bands. So quantitation of the band is very important. How we can quantify the band quantitation. Bands are anonymous. Because large number of bands we are generating with the so they are anonymously. So to identify a particular band, for significant band from the pool of the bands is very important. Now applications. Monitoring inheritance of agronomic trait, diagnostic of genetically inherited disease, pedigree analysis. forensic typing, identification of different hybrids, species level relationships, also in some cases at high level of relationships. Four, start code on target marker. Start code on target. Start code on target marker polymorphism analysis. SCORT is a novel method for generating plant DNA marker. Start codon. So as name self start codon means we have to look for polymorphism near the sequence of initiation codon. SCORT is a novel method for generating plant DNA. This method was developed based on the short conserved region which flanking the ATG start codon of the plant. Based on the short conserved region that is flank the ATG start codon. Okay, the G. Scott uses single 18-mer primer. This used 18-mer primer is for PCR and an annealing temperature of 50 degrees Celsius. PCR applicable are resolved using a standard electrophores. This method was validated in rice using genetic diagnosis set of the genotyping background operations. Here you can see Scott primer, the genes will appear development like this. Present absent, so it is a dominant marker. So here it's a representative of the different spot markers which are developed along with their sequences. So this is the spot profile of the two different parental genotypes that we do. Expressed sequence type EST. EST are the single pass sequencing read from randomly selected cDNA clones. Single pass cDNA sequencing reads they are randomly derived from the cDNA clone. EST are developed from the cDNA and cDNA are developed from the mRNA. So directly you can say express sequence tag represented a transcriptome profile of the and they are expressed parts as name itself expressed portion of the genome ESTs. Okay. It's identified through the cDNA. So how we can identify for the ESTs cDNA library? We can scan for the screening the cDNA libraries which contain many of the expressed genome. Pick the cDNA clone randomly. You can pick up the cDNA clones randomly. Rapidly determine some of the sequence of nucleotide from the end of each clones. These EST could then be compared to all known sequences using a program called BLAST. An exact match to a sequence gene means that gene encoding the EST is already known. If the match was close but not exact one, we could conclude that EST is derived from a gene with a function similar to that of gene. Then the EST sequence with the repetitive identification and approach in a gene bank and clone from various derived part kept in freezer for later uses. The overview of EST sequencing process. The clones are picked from the factory uses into microdata templates and achieved for later uses. All subsequent manipulations of PCR cleaner sequencing are carried out in microdata templates to yield medium throughput. This is the generalized outline of the EST sequencing protocols. So here are overall summary of these molecular markers with their comparisons. RFLP. RFLP, PCRFLP, RFPD. So detection method for RFLP is hybridization, PCR generally used, DFQ hybridization, RFPD PCR, microsatellite direct PCR. Type of proven primer, genomic DNA. RFLP uses genomic DNA, cDNA sequence specific primers, mini satellite. Case of RFPD we are using arbitrary design primer. Sequence specific primers. Requirement of radioactivity is not required. It is required also for, but in few cases not required. Extent of genomic coverage is limited in RFLP and PCR, while in case of others it is extension. So data analysis of SLPSR can be done with scoring of the different bands like 0, 1, 0, 1, 0, 1. What is present 1, absent 0. So likewise we can go for the scoring and we can analyze all these data. Similarity. So simple matching coefficient by measuring the proportion of shared band presence. Jakarta coefficient also proportion of shared band. Ning and Lee coefficient also by the probability of a band being amplified in one sample being unamplified in another samples. Okay, so major problem is false positive result is given RFID because it's a PCR based you can get false positive result also sometimes. So that is it. Whereas genetic similarity matrix is calculated about Jakarta coefficient based on molecular markers here, likewise get it. So there a lot of DNA marker application is there, fingerprinting. So DNA fingerprinting is generally used with help of RFLP, and by fingerprinting we can get the DNA profiling of different varieties, parental cases, different criminal cases. So fingerprinting helps give a DNA complete profiling of a particular variety also. And DNA fingerprinting is markers are useful. Diversity studies. So markers are very useful in the diversity. We are using different germ plasm or different diversity that is present in different germ plasm. So for that we can use the molecular markers extensively. We can use these markers. Then marker assisted selection. Obviously, marker assisted selection is based on totally molecular marker. As selection of a particular trait based on the molecular marker done by the help of molecular markers. Genetic mapping is also done with the help of molecular markers. Gene tagging. So tagging a particular gene is very useful whenever we are isolating a gene from a cell. Novel allele detections or exploitation of there is another term bioprospecting. In case of bioprospecting or novel allele identifications, markers are utilized. Map-based gene cloning is a method of gene isolation. Map-based gene cloning. If we know the genetic map, linkage map of a particular crop, so a gene can be easily cloned or identified. F1 identification or hybrid identification, you can say. F1 or hybrid identification can be done with help of well-characterized molecular markers. Then comparative mapping. Comparative mapping is suppose we are comparing the genome of two different species. So we can develop a comparative map that we can compare the different molecular markers while using molecular markers. We can prepare the genetic map for the two different species with bulk segregate analysis, seed testing also. So this is the vast application of the molecular markers. So this is all about the introductory lecture about the DNA and molecular markers. Thank you.